Hello everyone, welcome to Shel lectures. So in this video we'll be talking about biological classification, a class 11 NCERT biology topic. So what do we need to know about classification before actually getting into the lesson in detail? So since the dawn of civilization, there have been many attempts to classify living organisms. So there have been attempts to classify living organisms that has been taking place since the beginning of civilization. It was done instinctively, not using criteria that were scientific. So classification was not done scientifically during the olden times, but only so that we derive certain uses from the organisms for our own use, like food, shelter and clothing. They were classified. So what is the actually beginning point of this classification? So Aristotle, he was the first and the earliest person to attempt a more scientific classification, and he classified plants into trees, shrubs and herbs, and he classified animals into ones which had red blood and the other which did not. During Linnaeus' time, he proposed a two-kingdom classification. There is kingdom Plantae and kingdom Animalia, but this kind of classification did not distinguish between, that is, it did not differentiate the eukaryotes and prokaryotes, the unicellular and the multicellular organisms, and also it did not differentiate the photosynthetic, that is the green algae, and the non-photosynthetic fungi organisms. So why was this found inadequate? Why was it rejected at a later date? Because besides gross morphology, that is, these classifications were made just only by looking at the superficial morphological characters. That cannot be accepted when we talk about classification. We also need to include other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships and so on. So for a long time, different scientists have took different ideas on different times, and the classifications which they made were completely different from the other scientists. So all of these put forth a standard mode of classification which we'll be learning here. So this is the present day modern widely accepted 5-kingdom classification. There is Monera, Protista, Fungi, Plantae and Animalia. So their characters on the basis of which they have been divided are cell type, cell wall, nuclear membrane, body organization and mode of nutrition. So cell type for Monera, it's prokaryotic, Protista eukaryotic and all the others eukaryotic. Talking about cell wall, Monera, it's non-cellulosic, it contains some substances like peptidoglycan, and it's a polysaccharide plus amino acid. Protista, it's present in some of the protists. Talking about fungi, it's present, but it's present with chitin. Plantae, we know very well, cell wall is present and it's made up of cellulose. In Animalia, cell wall is absent. Talking about nuclear membrane, we can very well correlate this nuclear membrane with the cell type. So as monerans are prokaryotic, their nuclear membrane will definitely be absent, and all the others, the nuclear membrane is present because they are eukaryotic organisms. Also the body organization just increases as we go from the one kingdom to the other. So cellular, multicellular or loose tissue, organ system. And the mode of nutrition, for Monera, it's autotrophic and heterotrophic. For protist, it is autotrophic and heterotrophic, but only photosynthetic. Whereas for Monera, we have both chemo and photosynthetic. And for heterotrophic also, we have sapro and parasitic. Talking about fungi, it's purely heterotrophic. Plantae is purely autotrophic. Animalia is heterotrophic. There is holozoic or saprophytic mode of nutrition. So this 5-kingdom classification was actually proposed by R.H. Whittaker. So the main criteria, as we saw just now, is cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Also later, a 3-domain system was classified. That is, they just split kingdom Monera into 2 and the other were put in order. So if you look at if you think over the previous one which we learned, there were 5. So if Monera is going to be split into 2, that will become 2 and the remaining 4 will combine together to produce a 6-kingdom classification. So what are the issues that arose due to this system of classification? So earlier systems of classification, like all the organisms were put under plants only because they had a cell wall. So there was a common thought that organisms which had a cell wall should come under Plantae. So they included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and angiosperms, everything under plants. The character that unified is that they had a cell wall in their cells. So because of this, it brought together the prokaryotic bacteria and the blue-green algae and with the other organisms which were eukaryotic. So it did not actually distinguish between the eukaryotic and the prokaryotic organisms. Also, it also grouped together the unicellular organisms and the multicellular ones. For example, Chlamydomonas and Spirogyra. Chlamydomonas is uni and Spirogyra is multicellular, but they both were put together under algae. Also, it did not differentiate between fungi like heterotrophic group and the autotrophic green plants. Though fungi had a cell wall which was made up of chitin, and the green plants had a cell wall which was made up of cellulose, even then both of them were put into the same kingdom or same classification. So what were the changes that were made? So due to these controversies which arose, some certain changes took place. That is, all the fungi were put together in a separate kingdom which is known as kingdom fungi, and all the eukaryotic organisms were put under protist. Unicellular eukaryotic organisms were put under protist, and all the prokaryotic organisms like bacteria and all that were put under kingdom Monera. Okay, so kingdom Protista has now brought together Chlamydomonas, Chlorella. So all these were placed in algae within plants because they had cell walls, but now they are not there. Instead, they are in Protist kingdom. Also, they were put together with Paramecium and Amoeba. They did not have cell wall, but as they did not have cell wall in the earlier system of classification, they are placed in Animalia, but now they have been brought under Protist. So we need a phylogenetic classification. So we need to evolve a classification system which will not only talk about all these morphological and the internal characters, but also the evolutionary relationships between organisms along with physiological and reproductive characters of organisms. So first let us know about kingdom Monera. So whenever we hear the word Monera, immediately another word should come to our mind. That is bacteria. They are the sole members and they are the most abundant microorganisms also. And there is a fact that bacteria can live in any kind of extreme conditions like very hot areas and very salty areas. So all these kind of situations they can adapt to live only because of some certain characteristics that they possess. Also, they can live as parasites, and based on their shape, they are being divided into 4 types. There is the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrium, and the spiral spirillum, as you can see in the diagram in the next slide. So, in kingdom Monera, the genetic material is naked circular DNA. It's also called nucleoid, genophore, and prochromosome. Okay. So, genetic material is a naked circular DNA, genophore and prochromosome. Histones are absent, but other basic proteins like polyamines are present. And the ribosomes which are present here are 70S type. And the subunits we know very well for 70S type, the subunits are 50S and 30S. So, these are the shapes of bacteria: cocci, bacilli, spirilla and vibrio. So, the structure is very simple as you can see in this figure, but the behavior that they exhibit is very, very complex. So the various shapes of bacteria: coccus, examples Streptococcus and Staphylococcus. For bacillus, E. coli. For that is Escherichia coli. For vibrio, it's Vibrio cholerae, and for spirillum, it's Spirulina and Spirochaete. So on the basis of flagellation also we can split these organisms into various types, like A. trichus. Wherever the letter A comes, it refers to something which is absent. So A. trichus, that is no flagella. Monotrichus, single flagella, and the examples are Vibrio cholera and Pseudomonas. And Lophotrichus is a tuft of flagella is just found at one end. For example, Spirillum. And Petritrichus, which is flagella, will be present all around the body of the organism, that is E. coli. And Amphitrichus, the flagella will be present on both sides. So what is the nutrition? As we learned earlier, they are autotrophic, they synthesize their own food, and they may be photosynthetically autotrophic or chemosynthetically autotrophic. So, but majority of bacteria are actually heterotrophs. I told you that they are both autotrophs and heterotrophs. Bacteria are heterotrophs, that is, they depend on other organisms or dead organic matter for food. So, what are the economically important bacteria? Like nitrifying bacteria, Nitrosomonas, Nitrobacter, all these are chemoautotrophs, and they help in certain processes like ammonia with Nitrosomonas makes nitrite, and with Nitrobacter, nitrite makes nitrate. So, this is the complete process of nitrogen fixation, which we'll be learning detail in the further lessons. Also, Pseudomonas denitrificans causes soil denitrification. All these are bacteria and they are economically important. That's what we are learning under this topic. And there are also a lot of nitrogen fixing bacteria, and also retting of jute by the butyric acid bacteria like Clostridium butylicum is also taking place. And then curing of tea and tobacco by Bacillus megatherium. Also food poisoning called botulism. The process of food poisoning is called botulism, which is carried out by Clostridium botulinum. It's not Clostridium butylicum, that is the previous one. This is botulinum via botulin toxin. Photosynthetic bacteria are also present like chlorobium and rhodospirillum. They can perform their own like photosynthesis, and they can prepare their own food. And vinegar, which is formed by Acetobacter aceti, and it also turns wine sour. And food poisoning can also be done. Gas forming can be done. All these can be done with the help of bacteria known as salmonella, which is rod shape. So these many uses are there as far as bacteria are concerned. Then we have to know about archaebacteria. So they are very special because they live in most harsh habitats of the world. Like they can live in extremely salty areas like halophiles, extremely hot springs like thermoxidophiles, and marshy areas like methanogens. So what is the reason for their survival in these kind of areas? They have a different cell wall structure. That is predominantly the main reason for their survival in these kind of extreme conditions. Also, the methanogens which are present in the gut of ruminants like cows and buffaloes, they are responsible for the production of methane, a biogas which is like very useful in several fields. So this biogas is actually obtained from the dung of these animals. So they are living fossils, and the cell wall contains pseudomerin, and the membrane possesses lipids, carotenoids to protect it from harmful radiations. So all these are responsible for it surviving in very harsh conditions. So talking about methanogens, they are obligate anaerobes, that is they are not facultative. They can just live, they have to live in a host or else they can't survive. And they produce methane, which is used in biogas plants, and also they form cellulose in cattle. Talking about halophiles, they live in salt-rich media, and they are chemoheterotrophs, and they impart brownish color to the soil. Thermoxidophiles live in hot springs. Then eubacteria is true bacteria. They are actually having a rigid cell wall, and they also have a flagellum if they are going to be motile. So the cyanobacteria, which is also known as a blue-green algae, they have a characteristic pigment which is known as a chlorophyll A pigment, a universal pigment. So it is responsible for all the photosynthetic processes taking place in that cyanobacteria, and they are photosynthetic autotrophs because of the presence of chlorophyll A. So whenever these kind of bacteria are found in water, they are just surrounded by gelatinous sheath. Okay, so what happens? They just form like blooms. They bloom in polluted water bodies. They bloom. So cyanobacteria, we need to know about that. There is cyanophyceae or mixophyceae in other words. And there are unicellular cyanobacteria like spirulina, colonial like nostoc, filamentous like oscillatoria, and they all have pigments like chlorophyll A and phycocyanin. And they perform oxygenic photosynthesis like normal plants do, and these were the first known organisms on earth to liberate oxygen. This is important. And they also have a special structure in them which is known as heterocyst, which is responsible for nitrogen fixation. Some examples of cyanobacteria which fix nitrogen fixation are Anabina and nostoc. So we need to know about eubacteria. Their cell wall has peptidoglycan, which is composed of N-acetyl glucosamine (NAG) and N-acetyl muramic acid (NAM). So their cell wall may have mucilage layer called capsule or a loose sheath which is known as slime layer. And their cell wall also contains certain acids which are known as diaminopimelic acid, teichoic acid, and mycolic acid. Also, mesosome, which is equivalent to mitochondria which are present in cells. It's associated with respiration, and also also assists nucleotide division. So lateral mesosome and septal mesosome is there. Lateral mesosome helps in respiration, and septal mesosome helps in nucleoid division. And then we have a term known as chromatophores, which was termed by Smits. And with photosynthetic pigment, bacteriochlorophyll perform photosynthesis. So with certain kinds of photosynthetic pigments which are present, these perform photosynthesis, but chloroplasts are not present here. Okay. Then we need to know about plasmids, which are double-stranded rings of DNA. Extra chromosomal double-stranded rings of DNA, and they have some genes such as F factor, which is responsible for fertility and conjugation, and also we have R factor, which is responsible for resistance to antibiotics, about which we will be learning in detail in biotechnology unit. And also they have Call factor, and this term, these plasmids were discovered by Hale and Lederberg. So plasmids which are associated with nucleoid are known as episomes, which was found out by Jacob and Wohlmann. So this is nostoc, a filamentous blue-green algae which also fixes nitrogen, and heterocyst structure you can see there. Mucilaginous sheath can also be seen. And a dividing bacterium you can see there. There is binary fission. There is one organism splits into two, and each organism grows into a new one. So that is called binary fission, and each bacterium divides every 20 minutes. So what is the reproduction in bacteria? Binary fission, as we saw just now, and it has special structures known as endospores, which are present in anthrax, bacillus and clostridium. So just a small clue which we can have for this is ABC. And they are thick-walled spores which are formed during unfavorable conditions, and during favorable conditions they germinate to produce new individuals. Sexual reproduction like conjugation, transduction and transformation also happens in bacteria. Then talking about again eubacteria. So I told you that nostoc and anabina are some of the cyanobacteria which can fix nitrogen in specialized cells which are known as heterocysts. And what are chemosynthetic autotrophs? They actually oxidize inorganic substances like nitrates, nitrites and ammonia, and they use the energy which is released for their ATP production. Also they play a great role in recycling nutrients like the nitrogen cycle, phosphorus cycle, iron cycle and sulfur cycle. All of these can take part only in the presence of chemosynthetic autotrophs. Then heterotrophic bacteria, they are importantly decomposers. Also they are helpful in making curd, lactobacillus. Also they are helpful in production of antibiotics, penicillium, and they are also helpful in fixing nitrogen, nostoc. Also they are pathogens which cause diseases like cholera, typhoid, tetanus, citrus canker. All these are bacterial diseases which are caused by several pathogenic bacteria. Then we just now learned that bacteria reproduces by fission, but during unfavorable conditions they produce spores, about which we learned in the previous slide. There is endospores. They also reproduce by a sort of sexual reproduction. There is DNA transfer from one bacterium to the other. They also practice a kind of sexual reproduction. Also we need to know about mycoplasma here, which are pleuropneumonia like organisms, PPLO, which were discovered by Nocard and Rokes. So what is very special about them is that they don't have a cell wall. They actually they are the actually smallest living cells, and they can survive without oxygen. These three facts are itself enough to produce awe when we hear the word mycoplasma. So they are also pathogenic in animals and plants. So mycoplasma are known as the jokers of the plant kingdom because they don't have cell wall, and the cell membrane has cholesterol, but it's not present in other prokaryotes. But this, though being a prokaryote, contains cholesterol in its cell membrane. And mycoplasma also causes certain diseases like brinjal and witch's broom, and rickettsia. It's an intercellular parasite, gram-negative obligate intercellular parasite. It's actually an intermediate between bacteria and virus. It shows an intermediate character between bacteria and virus. For example, Coxiella burnetii, which causes Q fever, Rickettsia rickettsiae, which causes Rocky Mountain spotted fever, or typhi. There is Rickettsia typhi, which causes typhus, and Rochalina, which causes trench fever. Then chlamydia, we need to know about it. That is also a gram-negative intracellular parasite. It produces elementary bodies inside the host where it's actually present for reproduction, and it causes sexually transmitted diseases like STD and conjunctivitis like trachoma and psittacosis. So we need to know about kingdom Protista now. So all single-celled eukaryotes come under Protista. So we may have a confusion. As protists, some of them are also photosynthetic. A photosynthetic protist may appear to be a plant to someone else. And they have flagella or cilia, and they reproduce asexually and sexually by a process which involves cell fusion and zygote formation. And the 5 divisions or 5 classes which we learn under this kingdom are chrysophytes, dinoflagellates, euglenoids, limoles and protozoans in this PPT. So chrysophytes, first of all, they are diatoms and golden algae, which are commonly known as desmids. They are microscopic and they float in water currents. So what is special about them is that their cell walls are actually made up of silica, so they are very, very hard and they are indestructible. Also they fit together like soap box. So these diatoms, they leave behind a large amount of cell wall deposits, and they all accumulate and they form a thing which is known as diatomaceous earth, which is actually gritty. So this soil can be used for polishing, filtration of oils and syrups and all that. So diatoms are also chief producers in the oceans. That is, they are the chief producers in the oceans because of the very important thing known as diatomaceous earth which they produce. Then talking about dinoflagellates, they are actually colorful protists. They appear yellow in color, green in color, brown in color, blue in color or red in color depending on the pigment which is present in their cells. Their cell wall has cellulose plates and they have 2 flagella. One is longitudinal and the other is transverse. And very often, this is very important, very often red colored dinoflagellates, which are like example goniolacs, they undergo very rapid multiplication and they increase in numbers, and they make the complete sea appear red because they have red pigment, so they produce red color, so the whole sea becomes red. So toxins which are released by these kind of organisms may kill many aquatic or marine animals. Then euglenoids. Euglenoids are freshwater organisms and they are found in stagnant water. They do not have a cell wall, but instead they have a structure which is known as pellicle, and that is what is the reason for their flexibility. They also have two flagella, one is short and the other is long. And this is very important. They are mixotropic in nature. That is, they are photosynthetic in the presence of light and heterotrophic in the absence of light. They predate on other organisms when there is no sunlight, but when there is sunlight, they become photosynthetic. So they are both photosynthetic and heterotrophic, so they are known as mixotropic. So the pigments of which are present in these organisms are equal to some higher plants also. And very good example for this is Euglena, which we can even predict from the name itself. Then slime moulds, they are saprophytes, and the body actually moves along the decaying twigs and those garbage things. They engulf the organic material and they move on like that. So under suitable conditions, they form an aggregation which is known as plasmodium, and it may grow and it may spread to several feet. So during unfavorable conditions, the plasmodium differentiates and it forms spores at the tips, and the spore possesses true walls. So they are extremely resistant and they survive for many years, and then the spores are dispersed by air currents and they form new organisms. Then protozoans are heterotrophs, they are predators, they are parasites. So all these are negative as far as we talk about this. So there are four groups. Amoeboid, they live in freshwater, seawater or moist soil. They capture the prey by putting out a structure which is known as pseudopodia, which is false feet. So they just capture the organisms, their food. And a very good example for this is amoeba. They have silica shells, and a parasite under this category is Entamoeba histolytica, which causes amoebiasis. Then flagellated protozoan, the name itself says that they are flagella, and the parasitic forms causes some diseases like sleeping sickness. For example, for flagellated protozoan is trypanosoma. Protozoans, under that ciliated protozoans, they possess cilia and they are actively moving because they have thousands of cilia in them, and they have a cavity in them that opens to the outside of the surface. They have a gullet. The cavity in these organisms, like paramecium is a ciliated protozoan, so they have a gullet which opens to the outer surface. So the coordinated movement of rows of cilia. So what happens is the water laden with food materials are stored in the gullet. That is their cavity, the body cavity. And there are sporozoans. They have an infectious spore-like stage in their life cycle. Very good example for this is Plasmodium falciparum, which is a malarial parasite which is responsible for malaria, a disease which has staggering effect on human population. So the first image is dinoflagellates, the second image is Euglena, the third image is slime mold, and the fourth image is paramecium. So what about kingdom fungi? So the mushroom and the toadstools which we eat are also fungi, and the diseases which they cause. The white spots which we can observe on the mustard leaves, they are actually due to a parasitic fungus. Also some are useful, like yeast, they are used to make bread and beer. And other fungi like wheat rust causing puccinia, it's a very good example. Puccinia causes wheat rust, and some of them are good antibiotics like penicillium. So their body consists of thread-like structures called hyphae, and when a network of hyphae is found, it's known as mycelium. Some hyphae are continuous, that is, they are filled with multinucleate cytoplasm. They have a large amount of nuclei. They are known as cenocytic hyphae, but the others have divisions in between, septae, in their hyphae. The cell walls of fungi are composed of chitin, as we learned earlier in the very first slide. Most of the fungi are heterotrophic. Almost everybody of them are heterotrophic, and they absorb organic matter from dead substrates, and hence they are also known as saprophytes. And they are also parasites because they depend on living plants and animals. So parasites are those which depend on living organisms, and saprophytes are those which depend on dead. So they can also live as symbionts. A very good example for this is with algae as lichen, and with roots of higher plants as mycorrhiza. So this is mucus, first one, the next is aspergillus, and the third is agaricus. So all these are examples for fungi. So we need to know about ray fungi, which is actinomycetes. So they are actually mycelial mycobacteria. Many antibiotics are obtained from this. That is what is important here. Streptomyces griseus, an organism, it gives us an antibiotic known as streptomycin, which was isolated by a man known as Waxman, who gave the term antibiotics. And from Streptomyces erythraeus we get erythromycin. Streptomyces venezuelae we get chloramphenicol. From Streptomyces aurofaciens we get tetracycline. And from Streptomyces rhimosus we get terramycin. From Fradiae we get neomycin. So all these are antibiotics which are derived from different actinomycetes. And then we have also have pathogenic. Any organism which is producer of antibiotics is also going to be pathogenic. So they have cell wall which is rich in mycolic acid, and they cause TB, tuberculosis, and they cause leprosy. And the organism which causes tuberculosis is Mycobacterium tuberculosis, a well-known fact. And leprosy, which is caused by Mycobacterium leprae, and diphtheria is caused by Corynebacterium diphtheriae. So what is the reproduction? The reproduction takes place by fragmentation, fission, and budding. Asexual reproduction by conidia or sporangiospores or zoospores, which are motile. And sexual reproduction is by oospores, esospores, basidiospores. All are spores actually. We have 6 kind of spores. For asexual, conidia, sporangio, zoo. For sexual, oo, aso, basidio. So these spores are actually produced in structures which are known as fruiting bodies. And the sexual cycle, it involves 3 processes. First of all, protoplasm fusion, there is plasmogamy, then nuclear fusion, which is karyogamy, and then meiosis, which results in haploid spores, which can give rise to new organisms. Then reproduction, fungus reproduces sexually. 2 haploid hyphae mating types come together and they fuse. When they fuse together in some fungi, immediately it results in the diploid cells. But in some other fungi like asomycetes and basidiomycetes, an intermediate stage is formed, which is known as dikaryotic stage, that is N plus N stage. Then later on it fuses to produce 2N, but intermediately it forms N plus N stage, that is 2 nuclei per cell. It's known as a dikaryon phase and dikaryon condition and dikaryon phase of fungus. Then after that the nuclei fuse and the cells become diploid. So the fungi forms fruiting bodies, there the reduction division occurs, and then haploid spores are formed. Then talking about phycomycetes, asexual reproduction takes place by zoospores here or by aplanospores. These spores are produced in sporangium, and the mycelium is aseptate and cenocytic. A zygospore is formed by the fusion of 2 gametes. So because of the fusion of 2 gametes, a zygospore is formed, and they may be isogamous or anisogamous. That is, the gametes which they produce may be similar or dissimilar. Very good examples are mucor, rhizopus, the bread mold, and albugo, the parasitic fungi on mustard. Then asomycetes, just now we learned, it's sac fungi. It's multicellular like penicillium or unicellular like yeast, Saccharomyces cerevisiae. And they are saprophytic, decomposers, and they are coprophilous. That's what is important here. They grow on cow dung. And the asexual spores are conidia. They are produced in conidiophores. And conidia produce mycelium when they germinate. Sexual spores are asospores. They are produced in asci. They are sac-like structures in which asospores are produced, and they are arranged in different types of fruiting bodies called asocarps. So asospores are produced in sac-like asci, and they are arranged in fruiting bodies called asocarps. Some examples are Aspergillus, Claviceps, Neurospora. Neurospora, it's used in biochemical and genetic work. So many members of this asomycetes like morels and truffles, they are all edible and they are also considered to be delicacies. Then talking about basidiomycetes. So they are mushrooms, bracket fungi, all these come under this, like rust and smuts, all that. Asexual spores are not found. They do not reproduce asexually, but vegetative reproduction by fragmentation is common. The sex organs are also not present, but plasmogamy, it happens due to the fusion of 2 vegetative cells of different strains or genotypes. And the resultant structure, as I told you earlier, they do not form diploid immediately. They form an intermediate stage which is known as dikaryotic phase, and at last it gives rise to basidium. And they are arranged in fruiting bodies which are known as basidiocarps. So common examples are agaricus, which is mushroom, ustilago, which is smut, and puccinia, which is rust fungus. Deuteromycetes, they are known as imperfect fungi because only the asexual characteristics of this type of fungi are known. When the sexual forms are discovered, they were put into some other higher category. So they were moved to like asomycetes and basidiomycetes, but they reproduce only by asexual spores like conidia, and the mycelium is septate and branched. Some of them are saprophytes and some of them are parasites, but majority of them are decomposers. They help in mineral cycling. Examples are Alternaria, Colitotrichum, and Trichoderma. So then kingdom Plantae, it includes all the eukaryotic chlorophyll containing organisms, commonly called plants. So we know this very well. So the life cycle of plants, as we know very well, they include 2 phases like gametophytic and sporophytic. Gametophytic is haploid and sporophytic is diploid. They both alternate with each other. In all the plants' life, they have these 2 phases which alternate with each other. So the lens are actually varying among different groups in plants. This phenomenon is known as alternation of generation. Some of them are also heterotrophic, like insectivorous plants like bladderwort, Venus flytrap. So all these are examples for insectivorous plants, and Cuscuta is a very well known parasite. We'll learn about this in detail in the next lesson. Kingdom Animalia, they are heterotrophic eukaryotic organisms which are multicellular and they do not have cell walls. That is a very characteristic feature of Kingdom Animalia. They depend on plants for food directly or indirectly, and they digest into an internal cavity. They digest their food in an internal cavity, and the stored food reserves as glycogen or in the form of fat. And the nutrition which they undergo is holozoic, that is they ingest food, by ingestion of food. Sexual reproduction is by copulation, followed by embryological development. So zygote forms and embryo and all that happens. So viruses. So this is actually a very complicated topic. Scientists have not talked about this, especially R.H. Whittaker's classification, which we talked about right now. He has not included viruses in any place out of that 5 kingdoms. So actually they are not actually considered as living at all. They are non-cellular organisms. They have a crystalline structure outside the living cell. So outside the living cell, they have a crystalline structure. Once they actually attack a cell, they take over the machinery of the host cell, and they take over the complete control of that, and they destroy and they kill the host. Outside the host cell, they are inert. They are like inactive. Okay, so they are obligate parasites. That is, they need a host for their survival. And then we have HeLa cells, the cell culture which was derived from a cervical cancer patient named Henrietta Lacks. Plagues are also formed by bacteriophages. Bacteriophage is a virus which infects bacteria on bacterial cultures due to lysis of host cells. So as the host cells are killed by these bacteriophages which infect the bacterial cultures, plagues are formed, and the smallest virus is the mouth virus of cattle and foot virus of cattle, and the largest virus is pox virus like variola and vaccinia. Then the 4 types of viruses. They are double-stranded DNA virus. Most of the animal viruses like bacteriophages, cyanophage, adenovirus, pox virus, cauliflower mosaic virus, CMV, all come under this. Then we have single-stranded DNA virus like Cyan-2-174, double-stranded RNA virus, Rio virus, wound tumor virus, single-stranded RNA virus like most of the plant viruses are RNA single-stranded, rhabdovirus which causes rabies, arbovirus which causes dengue, poliovirus which causes polio, paramyxovirus which causes mumps and measles, or myxovirus which causes influenza, and tobacco mosaic virus, in short TMV, which is of 300 into 200 nanometer size. So retrovirus and oncogenic virus is what we have to know next. So retrovirus is actually a single stranded RNA virus, and it possesses an enzyme which is known as reverse transcriptase. So there is a process which is known as reverse transcription, which forms cDNA from this template, from this single stranded RNA. They form cDNA, and it was discovered by Tellman and Baltimore. Retrovirus was discovered by Tellman and Baltimore. Example, HIV is a retrovirus, Rho sarcoma virus, and then oncogenic virus is cancer causing virus. For example, hepatitis B virus, Epstein-Barr virus, herpes virus, human papilloma virus, HPV. So we need to know about virions, cyanophages, and transduction. Virions are mature active virus particles. Cyanophages are cyano. The word itself says B, something which is related to BGA, blue-green algae. They are the viruses which feed on blue-green algae, and it was discovered by Saferman and Morris in an organism which is known as Pectonema. It's also a BGA. Then transduction. What is transduction? It is a process in which the transfer of genes from one bacteria to the other via bacteriophages takes place. It was discovered by Zinder and Lederberg. So you can view the diagrams of tobacco mosaic virus here and bacteriophage here. The diagram of bacteriophage is very important, with parts like head, collar, sheath, and tail fibers. Also, tobacco mosaic virus, it consists of RNA and capsid. Tobacco mosaic virus is a plant virus, that's why it consists of RNA. Then bacteriophage, just now we saw parasites on bacteria. It was discovered by Tuart and Herrill, and the name was given by Herrill. The genetic material is DNA because it's something which infects animal cells, so dsDNA. Examples are T2 bacteriophage. It has a tadpole-like structure, as you can see here. And then they can have 2 types of cell cycles. They are lytic cycle and lysogenic cycle. Lytic cycle, it results in the lysis, that is the death of the host cells. Wherever they are present, maybe it may be bacterial cultures. So when they are present there, it results in the lysis of the bacterial cultures. So it is actually found in T2 bacteriophage that we saw just now. This is lytic cycle. What about lysogenic cycle? It's found in lambda phage. Here, multiplication does not involve the killing of the host cells, but the viral genome alone gets integrated with the host DNA and it forms a prophage. Okay, then it may change into a virulent phase on environmental stimulation. This does not cause the lysis of our cells, the death of our cells, but it actually undergoes a very gradual process. Then the history of viruses. The name virus, which means venom or poisonous fluid, it was given by Dmitry Ivanovsky in 1892. And how did he find this? He was the one who actually discovered the tobacco mosaic virus, and he discovered that these viruses are ones which infect the tobacco very hard. And then these were found to be smaller than bacteria because they could pass through the filter, the bacteria proof filters, so they were smaller than bacteria. And a man known as Bijenic, he demonstrated the extract. So I told you that tobacco mosaic virus infects tobacco. So he took the extract of tobacco and he tested it, and he got a fluid which was like very infectious, and he called the fluid as Contagium vivium fluidum. It is an infectious living fluid, and he named it like that. Who named it like that? Bijenic. And then we need to know about Stanley, very important. He was the one who performed the crystallization of viruses. He said that viruses could be crystallized, and the crystal consisted largely of proteins. Also, we need to know some more. Like, in addition to proteins, viruses also contained, as we saw now, RNA or DNA. RNA for plants and DNA for animals, but not both. A virus is a nucleoprotein, and the genetic material is infectious. So, plants, they are single-stranded RNA, and animals, they are single or double-stranded RNA or double-stranded DNA. Mostly double-stranded DNA. So bacterial viruses, just now we saw they are double-stranded DNA, and the protein coat is known as capsid. It's made up of subunits called capsomeres. It protects a nucleic acid, and they are arranged in helical geometric forms. So the protein coat is called capsid. So the diseases which are caused by viruses are mumps, smallpox, herpes, influenza, AIDS. It's a very, very dangerous disease which is caused by human immunodeficiency virus. In plants also, like mosaic formation, leaf rolling, curling, yellowing and vein clearing, dwarfing, stunted growth, all these are happening due to the viral infections. Then viroids. So something is absent, right? So here, it does not have the protein coat. Only that's the difference between this and viruses. Okay, it was actually only RNA. It contained only RNA, and it was discovered by Theo Diener, and it also had a very low molecular weight. So it was smaller than virus, and the disease which it caused was potato spindle tuber disease. Then prions, which was discovered by Prusner. So in modern medicine infections, we can actually found that neurological diseases were transmitted by an agent which consisted of abnormally folded protein. We cannot actually guess what kind of arrangement it has. Okay, but the structure is similar to viruses, and these agents were called prions. So diseases are very important. They cause bovine spongiform encephalopathy, called mad cow disease in cattle, and also its analogous form which is called C.R. Jacob disease in humans, CJD. It's commonly called CJD. So bovine spongiform encephalopathy or mad cow disease in cattle, and C.R. Jacob disease in humans. So prions are the causative agents for scrapie in sheep and goat, mad cow disease as just we saw now, and Kuru, which is laughing death, a disease of laughing death, and Creutzfeldt Jakob, that is C.R. Jacob, just now we saw, that is an analogy of mad cow disease. And Kuru, it was described by Gaj Dusak, and he was also awarded Nobel Prize for this. So then we need to know about interferons. They were discovered by Isaacs and Lindemann. So glycoproteins, these are interferons or nothing, but they are glycoproteins which are produced by horse cells. Whenever virus infects them, or whenever they come into contact with a viral infection, these interferons are produced so that, they inhibit the viral protein synthesis so that they can stay safe from viruses. Then lichens, as we learned earlier, they are symbiotic associations. That is the symbiotic association between algae and fungi. The algal component in lichen is known as phycobiont, and the fungal component in lichen is known as mycobiont. Algae is autotrophic and fungi is heterotrophic respectively. All these we have learned in the previous slides. So algae, what does it do? It's being autotrophic, it prepares food for fungi, and fungi being heterotrophic, it provides shelter, and it also absorbs lot of mineral nutrients and water for the algae. Their association is very, very close. That is, they have a very close relationship that if one saw a lichen, they could not actually guess that there are two organisms. They would come to a conclusion that there is only one organism. Also, they are very good pollution indicators. Wherever there is much SO2 polluted areas, they do not grow well in SO2 polluted areas. So, wherever there are no lichens found, you can easily guess out that that is a polluted area. So, that's it. The lesson comes to an end. So I hope you would have understood this lesson pretty well. Subscribe to my channel if you have not done so far. Like and share this video to whomever you know. Thank you so much for watching. I'll be meeting you with a lot of lessons and chapters in the future videos. Thank you.